So we're kind of a go-to facility.
The USF lab is unique because we have adapted cutting-edge cell biology tools. Tools commonly used to study the central nervous system, but really any type of cell in the body, to conditions that occur in adverse environments like diving. The dose of a gas that you receive is a function of two things: the percent that's in the mixture and the total pressure that that mixture's at. Air is like 80% nitrogen, and so you're fine at sea level. Now we go down 33 feet, it's twice the pressure. We go 66 feet, it's 3 times the pressure. You get down to about 100 feet, and it's 4 times the pressure. Then the dose is high enough, it's just like you're throwing a switch, and all of a sudden you act like you've had a martini or too many. In fact, they say it's like for every 33 feet you're going down, it's another martini. All right, so by the time you're at 100 feet of seawater, you're sloshed. All right, and you're going to do things like try to offer your regulator to the fish that are swimming by. Not a good thing.
Originally, the Office of Navy Research funded me to look at the cellular and molecular mechanisms of oxygen toxicity. And this led us to develop unique technologies that allowed us to look inside cells under conditions that would simulate, for example, a Navy SEAL diver.
Oxygen toxicity is seizures. It can happen within minutes as soon as you go beyond 2 atmospheres of pressure. We've been looking at 2 approaches. One is to understand the basic cell mechanisms that involved with oxygen toxicity, what makes the brain become hyperexcitable, that you see the seizure activity. And then we tend to ask a more broader question of, can we detect the onset of a seizure before it occurs, that one is impending, and can we delay it? And we've had good luck on both of those. One, we've been able to determine that breathing picks up several minutes before you go into a seizure. So we think that this is like a biomarker, a physiological marker of an impending seizure. Navy SEALs would breathe 100% oxygen because they wear a special device called an underwater rebreather. What this does is that they inhale and exhale right into the rebreather. So the gas is contained all within the breather.
We observed that the cells were overexcitable, that oxygen was a stimulus to the cells, and that metabolism was starting to break down. And this overexcitability led to increased hyperexcitability, which could lead to a seizure. So while studying CNS oxygen toxicity, I made an observation that was really serendipitous in that, first we observed that these cancer cells were kind of selectively vulnerable to high levels of oxygen and also being grown in ketones. A ketone ester is an alternative form of energy that the brain can use for fuel. And we found that cancer cells, basically, there was an increase in cell death when cancer cells were grown in it, and it also prevented their proliferation.
We found that this novel combination of ketosis with a ketogenic diet or ketone ester supplements and hyperbaric oxygen really dramatically slowed the tumor growth in this very aggressive model of metastatic cancer.
And we can also get the heart activity, the EKG, all simultaneously.
The ketogenic diet is a very low carbohydrate diet, and cancer cells, as we know from a long time of research that's been done, have a very high reliance on glucose, which comes from carbohydrate. So there's this idea that you could reduce the amount of carbohydrate in your diet and potentially slow the cancer's growth.